Hello everyone, Yamina here. Welcome to the Dr. GPCR podcast. Today, we're diving into a story that blends curiosity, serendipity, and science at its most inspiring. My guest is Alessandro Nicoli. He's a PhD student at the Leibniz Institute for Food Systems Biology in Munich, where he explores one of the most fascinating puzzles in our field, olfactory receptors. 400 unique GPCRs that mediate the sense of smell, yet remain some of the least understood. In our conversation, Alessandro shares how a birthday email changed the course of his career, why he refuses to pick a favorite GPCR, and how tools like AlphaFold and molecular dynamics are transforming the way we study these receptors. His journey is a reminder that sometimes the invisible can become visible if we have the right tools and persistence. Before we dive in, a quick note. Terry's Corner is now live. It's the only expert-led, on-demand pharmacology corner where Dr. Terry Kanakin breaks down complex concepts into practical insights. Each week, we release a new course and host live monthly Ask Me Anything sessions with Terry himself. This week's focus is on intracellular drugs. You'll learn how restricted diffusion compartments affect offset kinetics and rebinding, and how to define and calculate target residence time for better therapeutic coverage. You'll find the link in the description. And a big thank you to our partner, Celtaris Research. They're helping accelerate GPCR drug discovery with innovative chemical biology tools. For more details, check the links in the description as well. And now, let's dive into this episode.
Hello, everyone. This is Yamina from Dr. GPCR, and this morning I am very excited to have with me Alessandro Nicoli. Alessandro, welcome to the podcast.
Yeah. Hello, everybody. And thank you also for Yamina for this nice opportunity to talk with you and chat in this podcast.
Thank you. Thank you for coming. And I hope I did not mess up your last name.
Completely perfect.
Thank you. Thank you. All right. Welcome to the podcast. Very excited to chat with you. Why don't we start at the beginning? Could you please introduce yourself? Tell us who you are and what do you do?
Yeah. As probably I'm Alessandro Nicoli. As probably you can guess also from my accent or name, I'm originally from Italy and specifically close to Venice, Venezia. And I'm currently a PhD student in the molecular modeling group led by Antonella Di Pizio, the Leibniz Institute for Food System Biology that is based on the Technical University of Munich. And in my daily research, I focus on a variety of GPCRs that are called olfactory receptors. And outside the research side, I'm also involved in supervision of students and also tutoring and lecturing some lessons at the university. And this is, I would say, sum up of my daily life or introduction.
Thank you. Thank you, Alessandro. And I think if I understand correctly, Antonella was on the podcast. She's wonderful. We kept in touch since the podcast as well. And you and the lab itself has a very unique approach to study olfactory receptors. But before I ask you about that, I wanted to know, did you always know you wanted to be a scientist?
This is a good question. And I think I don't have a linear trajectory. I think probably as many of us. But I would say the first time that I think, okay, wow, it's so cool to do science. It was the university during my studies of pharmaceutical chemistry and technology at the University of Padua. And then I start, of course, with organic chemistry. And there, like the beauty of see the molecules, design the molecules, the reaction, thinking that you can create molecules was really exciting. But then, studying pharmacy, you get to know, for example, the biochemistry, and you see the reaction are applied also to how we live, how we breathe, how we move. And then I think the first point is when I encountered the basically medicinal chemist, and there was also thanks for a really great professor, Professor Moro, and then he helps a lot to see and open up a new world because the molecules that we knew how to make with organic chemistry was not only just a molecule, it was a partner that goes to interact with somebody else, and somebody else can be a protein, can be a DNA, an RNA, but has also biological meaning, and thinking to change small atoms somewhere can drive then to biologically effect that you cannot know. But the introduction of the other side, not only in the molecules, but also the protein and the interaction and the side chains, this one really got my fascination on the, basically to get to know more and more and say, okay, now I would like to really to get into deep into this topic, for example.
Yeah, no, I think you make a great point there. You always need a good mentor to get you interested in things, but at the same time, from a chemistry perspective, I find it fascinating that you modify the molecule to some extent, let's say a little bit, from a chemistry perspective, but the biological response is completely different, or you just get that different biological response. It's just fascinating. When I think
Sorry. No, I just think it's just the concept of the enantiomers. I mean, of course, just displacing on the space differently. You can have a molecule that is extremely potent, and the other one can be its antagonist, or can be less potent.
Dead.
This is really fascinating, and this is, I think, is the beauty of our world, or what we do in the daily life research.
Exactly. What got you into doing a PhD? So you mentioned getting interested in science as an undergrad, but then how did you end up?
Then, of course, I got to know, I say, okay, this is a topic that I'm really interested to. And I decided to pursue first the master thesis that, for example, in Italy is like between 6 and 7 months of research. And then I got to know for the first time, let's say, the second academic love, that is the computational chemistry. And there, basically, my, let's say, project was focused on drug discovery of a family of proteins that are the BCL2 family that are involved in many cancers like leukemia. And basically, we were trying to combine in molecular docking and NMR studies in order to get to know which molecules were binding and more or less where. And with the docking, we're trying to answer basically how the molecules can bind, just to recall what we were saying before. But then doing every day the research, I was getting in love with the topic, and decided, yeah, I want to do this in my life, let's say, and I want to continue pursue. And that one brings them to decide to do a PhD.
I love it. And how did you pick your PhD lab? How did you meet Antonella, for example?
This is a fun story, because after the let's say after my graduation, I started an internship studies at the lab where I was doing the master thesis. And the day of my birthday, that is the 15th of October, Professor Moro sent me basically the call of Professor Antonella Di Pizio, and I say, okay, let's try to apply here, because I was looking for a PhD. I was looking around, and then 2-3 weeks, basically we had the classical interview. We get to know each other. I think we really get a super match in first place, and then after a month, I was already in Germany. Basically in December 19, and there started my journey in the lab of Antonella. And also I was her first PhD. Basically we had like an entire lab and a view of to start and make her science and our science.
I love it, because I can relate to that. So I'm also an October born, but at the same time, when I started my master's, and a PhD, I started in a lab where there was nothing but me. I was the first PhD student, first master's student. There was just me and the PI. There was just bills everywhere and nothing else. So I can relate. It's when you're a young PI and your first hire matters a lot. So you pick them the right way, and you also have developed this extra relationship, I think, with your PI, because you're fresh, they're fresh, and there is an alignment in goals, which is to succeed and get publications. So you may get more attention than if you're a PhD student number 25 later on, because by then the lab is established.
Yes, I totally agree. I totally agree, and I can also relate on the words that you are saying. I mean, I remember my first day was like an office, and we were even sharing the desk because
Yeah.
then and then arrives the COVID, of course, that changed a bit. When we had more to change also our way to for her to mentor and for me also to learn.
Yeah.
But yeah, I can totally relate to what you were saying. And over the years, you see also the lab establishing and how, and for both of us, of course, growing up.
Exactly. So you mentioned how you discovered Antonella's lab. At the time where you were interviewing, did you know she was working on GPCRs? Did you find the topic interesting, or did you approach it from a computational chemistry standpoint and saying, well, this seems like a very difficult problem, I want to tackle it?
Let's say that even if, let's say in the lab where I was doing the internship, is also well known to study, for example, adenosine receptors. Let's say that I get to work a bit on molecular dynamics of GPCRs during my internship, and then when I start to see the membrane, the receptor inside, I say, okay, this is really cool. And also the fact that they can transmit the signal from above the cell inside and what they can cause is. And then when I applied to Antonella was the perfect match because it was computational chemistry, molecular modeling, and at the same time I can also dig on the GPCR field, even if it was a niche. Because of course, olfactory receptor, chemosensory field, that then we can focus a bit more then. But it was really a nice match between the challenge, I would say, but also the putting together, let's say, the molecular modeling and the GPCRs. But it was my first approach on the GPCR, letting apart the studies on the university, was during the internship, and then of course with Antonella had the greatest impact on the GPCR field and my focus on them.
I love it. Do you have a favorite olfactory GPCR?
Let's say this is a tough question in sense because they are 400. I mean, let's say that I cannot decide on them. Let's say let's embrace the challenge to study all of them.
Mm-hmm.
And it's really, let's say, choosing one specific is difficult, but they are beauty and they're unique because they can, let's say, they can bind many different molecules. Some of them are really narrow, but they're really interesting on this point of view on the selectivity, how they bind the other molecules. But I would say that I took the challenge to to all of them. I could not decide.
All right, then, and it's hard, and I completely agree. It would be very difficult to pick a receptor, and I push people to try and pick, but I'm not going to push you because there's 400 choices there. But I was wondering if you can tell us a little bit more about the approach or the questions that you're trying to answer through the work that you're doing in the lab.
Let's say that when I started, the world was without the impact of AlphaFold and so the impact of the structural biology. I mean, let's say that I started a PhD in this world, and then it's changed a lot during the years, luckily also with the novel technique. But let's say that other receptors, and more also to say also olfactory receptors, because they're also the trace amine associated receptors. That is another class of subfamily of class A GPCR that are these other 6 receptors, in total almost more than 400. And let's say that these the main challenge it is that there are few ligands known, and also the first structures were released last year, basically. And the main challenge was to work on the structural prediction of those receptors, and also to find any possible protocols that can lead to some new ligands. Because the ligands, because half of them are basically orphan receptors, and even if they have just one ligand pair, it's really weak, or anyway, they're small molecules, because other molecules, of course, are volatile molecules. And the challenge it was basically to get a face to those protein, basically the structure, to find a partners of them, and at the same time then can open up an entire world, because with the computer you can do so many cool things, and you can study how the activation, you can design new molecules to get more potent or more selective, but the main challenge it was that the information were really few, and they are really orphaned. So was the main challenge, I would say, the PhD.
Yeah, and how does AlphaFold help, or what did that change? Because you mentioned before, this is all before AlphaFold. Does it help at all? Does it help you do these pairings? And to your point, it's very difficult to work with receptors or any proteins where you don't know what their ligand is or what their interaction partner is. And if it's olfactory, it's volatile molecules, so it's very difficult. And how do you capture that? Even in the lab, it's very difficult.
Yeah. In fact, I just start from one of the, also in the not only computational side, also the experiment itself are really difficult, because you have problem of the expression, you have the problem that also to make the first picture of a mammalian odorant receptor, of course, was problem, because you need a ligands that stabilize that specific conformation, all the technologies that we know. And also from the, let's say, assay or structural biology point of view was difficult. But I think AlphaFold has, of course, as we know, revolutionized the world, as well the AI. And I think has been sealed during the last Nobel Prize, the Professor Becker and the developer of DeepMind. And just to say to the entire world that computational chemistry is there and is useful for. And in specific on the topic, it was surprising to see, basically, when they released the first structure of the odorant receptors last year, before AlphaFold already released the structure without any information of this. And basically, it arrives to have a match of really that are close to the experimental errors that you can have. I would say on the specific topic of the GPCR and olfactory receptors, I think the use of AlphaFold can open up a lot of the world, because now, let's say, the shift is not moved anymore to the structure prediction itself. Of course, they're not ready to use all of them, but now you have a plethora of 400 models that you can start with. Molecular dynamics, molecular docking, virtual screening. I think it's open up the world, and this has been confirmed by the experimental structures then.
That's fantastic. And to your point, you have a starting point. And let's add to it a reliable starting point. It might not always be true for all the receptors, but at least you know where to start from. I feel like it's when the first structure of rhodopsin was solved. We knew what it looked like, finally. And that also opened up the door. That didn't mean that we were able to solve all the other structures for the other GPCRs, or you can use the same. Because obviously, what I find fascinating about GPCRs, and I'm apologizing for rambling there here, it's a little bit early, and I went to the gym, but have not had coffee yet. That's next. But what's fascinating is that they all have 7 transmembrane domains. Most of them classically coupled to G proteins, yet they all modulate a variety of signals. And depending on what interactions they go through, either with the plasma membrane, with intracellular proteins, with the signals, with the extracellular ligands, they modulate a different physiological effect, which I find absolutely fascinating.
Yeah, absolutely. And it's also, I think now in this world, we can really, because we are this interconnection, like sequence, the structures and their dynamics. I think this is the most beauty that we can have and the most powerful tool that we can have to study our beloved GPCRs, because as you said, they can have these 7 transmembrane, but they can bind so many, can bind peptides, larger proteins, small molecules, odorant molecules. This is really fascinating. I think the main point of studying GPCRs is their beauty. They're similar. Let's say if you take a group picture of them, I would say they're kind of similar from distance. But when you zoom in, is just the beauty of their selectivity, of their action and function. I think is one of the main things that I got to love while doing my PhD and getting into the world of the GPCR. And more structures are coming, more studies we are doing, more questions are coming. I think we're just opening now the big door and getting into the real function of these receptors.
Wonderful. If someone who currently works in the wet lab and would like to get a better sense of what do you do as a computational chemist, what are the, let's say, top 3 things that they would have to read or dig into in order to get a taste of the work that you do?
Okay, this is a nice question because it can give also the possibility to give a bit of our work. Let's say that I would start probably with the work that we have done on a specific receptor called OR5K1. And basically, this is the name of the odorant receptor. They start with the family, OR5, and then they give some strange names and letters to nominate them because they are a lot. And basically there, we start from these receptors because we had, luckily, a set of active ligands and inactive ligands that were experimental, validated, and they're okay. We have at least we have solid, and those set was also nice because we're close related molecules. There were a little change between each other, and this was a really nice set to basically to test all our generated models. And then basically what we have done there, it was basically sample as much as possible with the computer let's say the binding site, when you're moving the side chain as much as possible, and iteratively computing all the rock curves, and you're basically assessing how good is the model to say, okay, this is an active molecules or this is an active molecules, and then to arrive and also pinpoint all the time with the experimental partners, for example, with mutagenesis studies or pre-knowledge on the sequence, and just focus more and more to some residues and to get then some let's say models that can be that can discriminate really well between active and inactive, and then you can use then this let's say pipeline to try to basically discover novel ligands, because again the problem, as we said at the start, it was we don't have ligands. If we don't have ligands, we need strategy to find new ligands.
Exactly.
This is the point. This is I think one of the start of our work that can help, for example, to dig into the how odorant receptor works, which is the binding site, and how basically to optimize, let's say, the model in order to predict novel ligands.
I love it. I love it. What's the I was going to ask it maybe in a naive way, but from a molecular dynamics perspective, in order to understand how does a receptor interact with one or a series of ligands, what are some of the questions that molecular dynamics can help answer?
Yeah, I mean, as all the computational technique, I think they can be used all together, or because I think that one specific questions can be answered to one specific technique or a group of technique. But for example, I think the beauty of molecular dynamics it is that you have, for example, as we said before, we have the membrane, we have the molecules of waters that shape our life and shape also the dynamics of the receptors. But let's say the power of the MD, it is that you can simulate over time, and the receptor moves in a physiological condition, and for example, you can put different ligands and see how your receptors basically moves. But at the same time, you can also basically simulate your receptors in different environment. I know you can add the cholesterol or point site mutation, and you see how this mutation can affect it's a stable GPCR, also for the lab, also to talk with the lab. And then, of course, if you go to the most fancy technique, like the group called metadynamics, that you start to push a bit the system with a bit of energy. There you can get to know also how this receptor get activated, or more difficult event, like, for example, because then we always study the ligands inside, but how they arrive there or how they get out, exactly. Is another point of view, because this is another big question that molecular dynamics can try to answer.
I love it. You mentioned that you also teach and mentor can you tell us a little bit more about these activities outside of your research focus, and how do you balance it all?
Okay, this is a really good question. Let's say the first 2 years, of course, they were COVID, and they were also for me training to learn. But then slightly, because for example, here at the TUM, we have basically we have not only the master thesis of bachelor, but we also have internship students that are for their curriculum, they have to come. Our lab is not only coming forming by PhD or postdoc, but we have also many internship coming for 2 months and everything. And let's say I also get to know how to handle these things. But let's say it's a really nice part of the job, because you have if you learn a bit how to manage people, because then you were focused on you, but then you have also you have other people that relies on you.
Yeah.
And but it's the beauty that you can work together, because then you're not only, because you have 24 hours, some you sleep and the other one you go to the uni. And basically there, if you can work together with 5, 6 different students, you can work through 6 different projects. You can move from ligand design, molecular dynamics really fast, and also different type of GPCR too. I would say this is the most outcome, and both of you grew up. I would say this is the nicest part of the mentoring, and also being able to talk with different people, and you grew up too.
I love it. I love it. And, so yeah, long story short, I was recently in a manager boot camp, and they were talking, one of the topics was, especially when you start off by, being in what they call an individual contributor, which people are for a very long time in a PhD or a master's or even a postdoc, and then you, let's say, go to a point where you have direct reports, you have people you're mentoring, but you're also responsible for. One of the difficulties is to delegate and to say, well, because you're the expert, right, at whatever you're doing. And in this case, computational chemistry and working on olfactory GPCRs, but then you need to be able to delegate some of these tasks to the people you work with, who work for you. How can you maybe tell me if you ever encountered a situation where it was difficult to delegate, or how easy is it for you to delegate?
No, let's say it's a process. It's a process that is still going on. It's not finished yet, of course.
It's never.
Let's say that I'm a rookie, not even a beginner of the mentoring. But let's say that at the start it was a bit difficult, honest, because you see all this project and you have to give to somebody, and you think, I don't know, but then I have to check thousands of times. But then with the time, as I said before, of course, it's a lot of investment, because especially at the start, is a lot of investment of time that you put on the person to learn the technique. And then, let's say, I think you can also adjust, because I think another thing that I learned over the time, that you can adjust some task also according to the interest of the person. Also this one, but of course you have to put a lot of time. I would say that you have to put a lot of time, and you have to have a check with them, because you make the point. I think the answer it is that you were making this mistake before.
Exactly.
In some of these mistakes, they have to do it too, because otherwise they will not learn. But you can guide, and you can guide them to learn, basically, and get to know basically the technique and go over it.
Yeah.
But at the same time, I think it's because you passed the same struggle before.
Exactly.
But the delegation, I think you have, or you do everything by yourself. It's an option, and this is an option. But again, it's 24 hours, and I think if you embrace and you say, okay, I can work in a team, I can grow up with the students, I think it's really, it's the most powerful things that we have. Also because you shape the new generation too. And there are more advantages, I think, a lot of time, but I think the reward is too big.
No, I agree. I love it. Thank you for that. All right, top 3 aha moments that you had so far in your career that shaped your trajectory.
Okay, but I would say, first of all, it was during the universities, and when, of course, Professor Moro get to know basically the application of Professor Antonella Di Pizio. This, I think, is one of the most point. Then I would say that also COVID shaped a lot, like the will and the strength, because of course for all the PhD, all the researchers all over the world, I think this was strengthened a lot of my motivation. Say, okay, if we can do it during with a computer in this room, because I was in the room mainly, we can also do it. And then I would say is a more generic, but the third point I would say the whole PhD trajectory so far, because I had the pleasure to work with really great people around the world, and this one really make me growing up a lot. And also, yeah, I would say the third point is also all the people that I encountered during the PhD that really shape also my vision and my way to do research. Also, because I mean, of course, my career is still small. I don't have big.
Well, we all have our aha moments. It can be at 5, 20, 25, or, during running an experiment and realizing something. It's not, you don't have to have a 25-year-old career. No. I think we all have that. And then, maybe in 5, 10 years, you can come back on the podcast and we'll have a chat, and then we can talk about different aha moments.
Yeah, this is I would say it is. I take already. I say already yes for the next 10 years. And
I love it. I love it. Any advice for scientists working in the field who want to get into the computational chemistry field in order to complement, let's say, their lab skills?
Okay, I would say that, I mean, as I said before, we are living in a really golden era for computational chemistry, and this is driven by the AI technology. It's driven by also all the good technique that are coming from the labs that develop basically software and algorithm for whatever, can be docking, molecular dynamics, or all the others. I would say that the first thing is the motivation and the passion. So I know that it sounds probably average to say, but without passion, you cannot go. But then another things that I really found really a bit more easier nowadays also to get to know the computational chemistry tools, because there are really a lot of tutorials. There are a lot of web servers. There are so much materials online, and that you can really get to know a lot of. And the third one, of course, is coding. For example, get to know Python, because it's really, I don't want to say mandatory, but it can help a lot. It can help a lot. That way, it's really satisfying when you see the computer working with the instruction that you provide, that they can give you back something. But I would say that the material online now, it's really big, and you can find really many knowledge there that you can really start to complement.
Mm-hmm. Any favorite materials, knowledge websites that you would recommend? Because as you said, if you go online and you Google it, there's so many ways you could go. But when you don't know what's best, then you ask the experts like you to tell us if you were starting today with the tools that are available to scientists today in computational chemistry, what are the ones that you would recommend starting with?
For example, there is a really nice, let's say, they're called tutorial. And basically, they're really nice because they cover all the aspects of the computational chemistry, but divided by basically, let's say, topic. For example, cheminformatics, that is branch of the computational chemistry that looks, for example, at the chemical structures of the molecules. And then for every of them, for example, you have a part of theory, and then you have the part of code explain. At the end, you have also some questions. This is a really nice tool, and you have for all of them. And the nice that they built also a story over it. And there is also a paper that I can share also the information with you.
Please, that would be great.
This can be a really nice, but also all the, I would say, many of the, let's say nowadays, of the package that you can use, they have they put really a lot of effort on the tutorials and the explanation of what to do step by step.
I love it. I actually was Googling tutorials as you were mentioning them. I will definitely take a look because I think I'd like to explore new things, but at the same time, this might be something we could do for Dr. GPCR as well.
Yep.
on the biology side.
Yeah,
and
we can
even on the computational chemistry side, that could be really awesome.
Yeah, we can have some resources that, I mean, they're there. Sometimes it's just to have the keywords to find this material online, let's say.
Awesome. No, anything you can share with us, that would be fantastic. And I think it would be really useful for the community, but I'm looking forward to digging into it. I just clicked on one of these very quickly. I'll take a look after the podcast.
Perfect.
Fantastic. Last question, the most difficult of them all. What should we title this episode?
It's a really tough questions, but let's say like the flavor of research and computation. If I just put together the two words.
Computational flavors of research of olfactory GPCRs, maybe?
Yeah, also can help. Also, yes.
Computational flavors of olfactory GPCRs. I love it. I really love it. It was simpler than, it's always a big question for me is what do we title the episode? But I think this is really good. Awesome. Alessandro, thank you so much. I really enjoyed our conversation.
Yeah, I really enjoy too. I would say also thank you for everybody that will listen to this podcast. And thank you, Yamina, also for the work that you put on the Dr. GPCR side.
Thank you so much. I really appreciate it. Don't go anywhere. We're going to stop recording, but I want to get a sense of how it went after we stop recording. And for those of you who are listening, you'll be wondering, what else are we talking about after we stop recording? Thanks, everybody. Thanks, Alessandro.
Ciao.
Huge thanks to Alessandro Nicoli for joining us today and sharing his journey from pharmaceutical chemistry in Padua to decoding the molecular dance of olfactory GPCRs in Munich. What struck me the most was his reflection that molecules are just static entities. We know this. We know they're partners in dialogue with proteins, DNA, and receptors. It's a perspective that reframes chemistry as a living bridge to biology, and one that can inspire each of us to look at our own science with fresh eyes. If this episode resonated with you, make sure to subscribe to the Dr. GPCR podcast and share it with a colleague. Leave us a 5-star review on Apple Podcast and subscribe to our channel on YouTube. You can also join the Dr. GPCR ecosystem to connect with collaborators who share your passion for discovery. Premium members gain direct access to a lot of perks. Check out the details in the description. And don't forget to dive into Terry's Corner. With 4 new courses each month and live Q&A sessions with Dr. Terry Kinaken, it's where you can sharpen your own pharmacology skills. This week, you'll compare intracellular versus extracellular receptor engagement and explore how intracellular access enables targeting novel allosteric sites. Plus, check out our featured partner, Celtaris Research. Links are in the description. As always, thank you to the entire Dr. GPCR team for making this episode possible. Stay connected, stay curious, and keep pushing science forward. Until next time, stay safe.